wish to thank the Academy for giving a chance to opportunity to talk here. And very good afternoon to all of you. And coming back right after lunch. So, can I have the lights then? Oh, thanks. So I'm kind of going to give an introduction for the non-biologists. So biologists, bear with me. A short introduction. First, I'll talk about what is gene regulation. So as many of you may know, even if you're not biologists, a newly fertilized egg to develop into a baby and the baby to grow into an adult and for the adult to function, the required instructions are in the set of genes that we get. And interestingly, these set of genes are present in all cells of, more or less all cells of our body. But then how brain does what brain does and not what intestine does and vice versa. And that is because not all genes are active in all cells at all the times. So their expression is regulated in terms of where and when and how much expression will be there. So that's what we call as gene expression regulation or simply gene regulation. And there are many mechanisms of regulating gene expression. And a group of molecules involved in them are small RNA molecules. And today the talk is about one component involved in the small RNA-based gene regulation. Okay. So there are three major classes of small RNAs. So the small RNA meaning these are single-stranded RNA, about 21 bases to 26 bases, majority of them 21 to 22. And there are three major classes. One is miRNA pathway. So the small RNA involved in this pathway are the microRNAs or miRNA. This is siRNA, short interfering RNA, and then your piRNA. So my talk is going to be primarily on this. So all of them involve a small RNA segment that helps this complex called RNA-induced silencing complex to find the target. The sequence specificity of identifying the target is what these small RNA molecules specify. And this RNA protein complex is essential to execute the function of preventing the gene expression largely. There are different mechanisms of preventing gene expression that I'm not getting into. So miRNA are produced by our own set of genes, the endogenous, and their function is primarily to regulate gene expression during development and as well as in normal adult function. And siRNA defends our body from pathogens like virus, viral RNA or viral DNA, any foreign genetic material gets silenced by this pathway. The piRNA based silencing protects the genome of our germline. So when I say genome, I mean the complete set of genes present in one particular cell. So when you talk about germ cells, these are the cells that give rise to egg in females and sperm in the males. And their genome needs to be specially protected because the survival of the species, like the integrity of the genome in the next generation, depends on the integrity of the genes in the germ cells. So therefore, germ cells mutations or invasion by foreign genetic element is not tolerated. So they have a special protective mechanism. That's where the piRNA-mediated gene silencing plays a role. So in this work, I'm going to show you some evidence where we have identified a protein that we believe is part of this in mediating silencing in the germline. Okay, so that's the summary of my talk. So we do all of this using this wonderful model organism called Caenorhabditis elegans. It's a free-living nematode that you usually find in temperate conditions in rotten vegetables and fruits. This is the head to tail. It's about 1 millimeter, so you need a dissecting microscope to work with. So it's not this big a worm. And what's highlighted here is the gonad. So this is where the mitotically proliferating germ cells, then as they differentiate and mature as gametes, they progress in this direction. These are the oocytes and these are the embryos. So you have two gonadal arms. From both sides you have oocytes coming, and then here you have the embryos. And I'm able to show all of this because the cuticle of the worm is transparent. You can see through it. And that helps in recovering mutant phenotypes very readily. So in the interest of time, I won't get into the details. And it's very easy to get the gonad out. If you cut here with a needle, the gonad will come out like a nice long tube. Okay, so this looped structure is what is straightened here. The loop portion is here. So here you have the different stages of germ cell development spatially arranged. Sorry. So here you have the mitotically proliferating cells, here progressively in different stages of meiosis, and these are the oocytes and these are the embryos. Okay, so I'll skip these. Okay, so in a genetic screen, a former graduate student, Lenita Thomas, discovered an interesting phenotype for a mutant in a gene that that's called PLP-1. I'll explain what it is in the next slide. Here the focus is on the mutant phenotype. So this is the gonad, the same structure as this, stained to visualize the chromatin of the germ cells. So each one is a nuclei here, what you see here. These are the oocytes. And in the PLP and mutant, the mutant here meaning it's not partial loss of function, it is complete loss of the protein coding part. So it's a null allele. When you grow at 20 degrees, that's the temperature at which we grow the worms in the lab generally, it's quite normal. It makes oocytes. Okay, it's like wild type. And when you grow at 25, the wild type worms are fine, but the PLP do not make oocytes. They only make sperm. Sometimes you do have mitotically proliferating cells where actually sperm should have been. And this is the phenotype she found, and this was interesting for us because we are always, as the introduction mentioned, we are interested in finding new genes involved in germ cell development. Okay, so we focused on this. So now what is this PLP-1? PLP-1 stands for per alpha like protein 1. What is per alpha? These proteins in other organisms originally shown to find bound to purine rich single stranded nucleic acids, DNA or RNA, and that's why they named it as PURA. And in C. elegans, they named it as PURA-like protein, so it is PLP-1. So para-alpha was an intermediate name. Now people focus on using this. So this is present in all organisms, from bacteria to human. Okay, and in vitro studies have shown, and cell culture studies, that it is found in RNA protein granule structures. So this is an alignment showing the relationship between the C. elegans PLP-1 and the human para-alpha. So there are conserved domains, and I'm not getting into that for today's part. So before us, people have done a knockout in mouse, 2 different groups, and they found problems with the brain development. So brain is, it's a section that's very enlarged, and this is a protein in the neurons that aggregates in the mutants, that's not in the wild type. And later people discovered through whole genome sequencing, some of the children born with neuronal abnormalities actually have mutations in different parts of this PURA protein, and these are the visible symptoms, and these children do not survive to adolescence. So it's a sporadic mutation. Before reproductive maturity, the child is dead. Okay, now increasingly more and more number of patients are being identified with this. Almost every month I read 1 or 2 papers describing a new mutation in human. Okay, so now getting back to our own story. So once Lenita found the phenotype, she decided to look at where it is expressed using antibody as well as a endogenously tagged fluorescence reporter, that is GFP. In both we found that it was present in the throughout the germ cells, okay, in the germ line, in all the germ cells we see it. And when she took a close look, it is found on granule-like structures that is around the nucleus in a perinuclear fashion. And when she used a marker for the nuclear envelope, she finds this is outside the nuclear envelope, it doesn't co-localizes with. And people do know RNA protein complexes present in this fashion already. And for example, one of them is PEGL1. We did a co-localization and that perfectly co-localizes, indicating that PLP-1 is on those germ granules present in germ cells. And it's there in the embryo as well. In 2-cell embryo itself, the germline components are segregated into the one of the two cells, that's always the smaller one. So that is where we found the PLP-1 granules, and some of them co-localize with the germ granule marker PEGL1. So then later a new student, Nihar, he looked at the other regions where we, Nilinitha had originally seen the expression. Like in the head region, it's found on the neurons as well as in the tails and in the ventral nerve cord as well. So it's present in germ cells as well as neurons in granule structures. That's consistent with what people have observed in cell culture studies where it is on RNA granules. Okay. So then Vishnupriya started to look at what does it do, and one of the things that we normally do is we focus on translation regulation. So therefore we have reporters in our lab where the reporter expression is governed by the 3' untranslated region. So much of the translation regulation of the germ cell germline is via the 3' untranslated region. So we have a bunch of lines and she looked at them, and in one of them what she ended up finding was in PLP-1 mutant, this particular transgene, PAL1 UTR driven transgene, is expressed in the oocytes while it is not expressed in the wild type. So immediately we would think that PLP-1 regulates translation via PAL1 UTR. But that's not the case, because wild type worms, when grown at 25, where germline gene silencing is defective, you see the expression there. So essentially what is happening is, for the silencing to happen at 20 degree, PLP-1 seemed to play a role. So this is what we suspected, therefore we wanted to investigate and find whether that is true. So we looked at bunch of genes that are known to be silenced in the germline. Initially itself, remember I told you there is a special mechanism in the germline that protects against foreign genes. That mechanism does silence some of the endogenous genes as well in normal conditions, that is 20 degree. So some of the transgenes, for example this GAP gold one, is normally silenced in the wild type, and when we introduce into PLP-1 mutant, it is expressed very nicely. And another transgene we tried, these are integrated single copy transgenes, and that is also expressed. So this again was consistent with our original idea that transgene silencing seemed to require PLP-1. Then we looked at some extra chromosomal transgenes. So these are when you inject plasmid DNA with a promoter and a coding sequence and a reporter. These plasmids form a tandem array outside the chromosome and get transmitted in a non-Mendelian way. These will express nicely in the somatic cells, but they have telltale marks of foreign genes, so they get silenced in the germline. So one of them is this LET858 GFP. So these are the gonadal cells, somatic cells. So these are the gonadal somatic cells. There you see nicely the GFP, but the germline GFP is completely off. Whereas in PLP-1 mutant, in addition to those gonadal sheath cells, you have germ cells also expressing. So for integrated transgene, you need this for silencing. For extra chromosomal array also, you need PLP-1 for silencing. So next we decided to look at whether the PIRNA-mediated, which the PIRNA or the small RNAs that are in core to suppress the transgenes and foreign genes in the germline, including transposons. So we wanted to test what happens to PIRNA-mediated silencing. So one group had published a PIRNA sensor where they have the promoter that drives expression in the germline, and GFP is our reporter. This helps in visualizing because this takes GFP into the nucleus, and this UTR doesn't have any translational control activity. So in this, when you put a PIRNA target, so this is the complementary sequence to a given PIRNA. So in the wild type, that PIRNA will bind here and suppress it, and when Vishnupriya did this, so this is example to show what to expect. Like this paper has shown in PRG and another gene involved in this pathway, the silencing doesn't happen. In the wild type, it is neatly silenced. So here PIRNA based silencing mechanism is defective, that's why it is expressed, and that happens in our thing too. So this is one more example, another piRNA we tested, and in both we do see it in the germline. Ignore this, I forgot to tell you earlier, this is autofluorescence of the gut granules. So these are the germ cells. So next we wanted to look at whether it is required for initiation of the silencing or for the maintenance of the silencing. So for that, if you introduce an already silent transgene into PLP-1 background, then if it is required for initiation, then it will remain silenced. If it is required for maintenance, then of course in the absence of it, then it should come, it should get turned on. So what we found is it doesn't get turned on. So this is a control wherein this mutant which is required for this protein is required for maintenance, and you do see robust expression. So this we repeated for all the 3 classes, and we found in all cases it's required only for initiation but not for maintenance phase. So, so this is where we stand. If I have a couple of more minutes. So then we wanted to get into the mechanisms, and Andy Fire and his postdoc Lamia helped us to sequence the small RNAs to see whether small RNA biogenesis itself was affected in PLP-1. So they found that they are not affected. So this data is from them. And then we looked at all the mRNAs, like we did transcriptome profiling. And transcriptome profiling means essentially you sequence all the mRNA in the 2 different conditions, in this case wild type and mutant, and look at which mRNA is increased or decreased in your mutant in comparison to the control. We found 2 groups, collagen related and sperm related genes are significantly upregulated. In total we found 780 mRNAs are upregulated, some 589 downregulated. So we are trying to tease apart what is required and why they are upregulated, etc. So this is validation of this whole genome sequencing result. So that I will skip. So another strain evidence to support our conclusion that PLP-1 is required for germline silencing is we looked at the genes upregulated in other pathway components like PRG1 that I mentioned earlier and MUT16 another one. So the upregulated groups largely overlap. So that's what these Venn diagrams tell you. And among them, predominant group is the sperm related genes. 96% of the sperm-related genes upregulated in PLP and mutant are upregulated in both PRG1 and MUT16 as well. So in summary, I have told you that PLP-1 acts downstream of small RNA biogenesis because Andy and Lamia showed it's not required for that. And we have shown that it is for the initiation, not for the maintenance. And from the 2 introductory slides, I showed you about mouse and human data, and we kind of think that this is not specific to germline gene silencing. PLP-1 probably plays a role wherever RNA protein granules regulate gene expression. So that's what we are trying to figure out currently in the lab. So with that, I'll thank the funding agencies and thank you all for your attention.
Okay, time for one quick question.
Yeah.
How good is the homology between MI, and pi?
Oh, so only the size-wise they are similar, but sequence-wise no, because these are each one have their own specific targets. So piRNA are tens of thousands of them. They pretty much cover the entire genome. And you have hundreds of miRNAs, each one specific for a given mRNA, or a group of mRNA at least.
So the piRNAs are the most diverse in terms of sequence.
Correct, correct.
We have time for another quick question.
Yeah. Well, ongoing experiments suggest so. Probably next time I'll tell you for sure. Yeah.
So are piRNAs expressed only in the gonads or?
Yes. piRNAs were originally discovered in germ cells, but I can't say certainly that it is not existing elsewhere. It's that people have not found yet.
All right, so let's thank Professor Subramaniam once again. So our next speaker is Dr. Navin Bist. Okay, time for one quick question.
Yeah.
How good is the homology between MISI and pi?
Oh, so only the size-wise they are similar, but sequence-wise no, because these are each one have their own specific targets. So piRNA are tens of thousands of them. They pretty much cover the entire genome, and you have hundreds of miRNAs, each one specific for a given mRNA or a group of mRNA at least.
So the piRNAs are the most diverse in terms of sequence.
Correct.
We have time for another quick question.
Yeah. Well, ongoing experiments suggest so. Probably next time I'll tell you for sure. Yeah.
So are piRNAs expressed only in the gonads or?
Yes. piRNAs were originally discovered in germ cells, but I can't say certainly that it is not existing elsewhere. It's that people have not found yet.
All right, so let's thank Professor Subramaniam once again.